Hi, my name is Brendan Carson. I'm a graduate student at Tufts University in Boston. And in this presentation, I'm going to use larval butterfly occurrence data and a model of larval dispersal movement to show that shared conspecific habitat preferences can influence the relationship between host-host contact and population size. This has the potential to increase disease transmission rates, substantially lower the population size required for pathogen maintenance, and create stronger than expected density feedbacks. Baltimore checkerspot butterfly is a species that's found in eastern North America. And originally it was a wetland species and was reliant on turtlehead or Chelone glabra, which is a wetland plant. But relatively recently, I think in the 1900s, many populations in the Northeast have made a shift to using Plantago lanceolata as their primary host plant. And we think this shift was really facilitated by shared chemical traits, mostly iridide glycosides that are found in both plants. But really what this has meant is that in many places, this butterfly is now found in drastically different types of habitat than they evolved in. So you can find them in dry old field sites now. And very recently, just in the last few years, it was discovered that there is a novel densovirus that affects this butterfly. So this virus is known as the Geonia-Senia densovirus, and it was originally found in buckeye butterflies, Geonia-Senia. And but recently, it's been detected at 14 out of 14 sites that this lab group out of University of Nevada, Reno has looked for it in. That's the Smilanich lab group led by Angela Smilanich. And while we don't know yet whether it's actually causing population changes at these sites, this virus definitely causes mortality both in the caterpillar and larval or in pupal stages. And it's been found on host plants, and we think it's primarily spread at that larval stage. Transmission is mediated by contact between hosts, just generally in most pathogens, most diseases. And in non-vector diseases, transmission is expected to be proportional to the contact rate. Spatial patterns in host plant abundance and quality influence herbivore contact rates and resulting pathogen transmission. And so these female butterflies are choosing where to lay their eggs across the site. We don't really know exactly what's driving this. It could be host plant density, host plant chemistry. They could be trying to decide whether the site is similar to a wetland where they're laying their eggs, or it could be that floral abundance actually draws them into a particular area. But in any case, these decisions lead to the spatial pattern of larvae on the landscape. Most epidemiological models assume a homogeneous spatial structure. One feature of this assumption is a direct linear relationship between population size, density, and the population threshold required to maintain a disease, known as R0. However, the way animals use habitat often has a spatial structure that influences how density interacts with population size. This has the potential to influence the minimum population size that will allow a disease to persist. The top set of panels shows a species that uses available habitat evenly, while the bottom shows a species that clusters in high quality habitat. Notice how these different spatial distributions change the population size required to maintain pathogen persistence, shown by the red line. Using the checkerspot system, I'm going to address the following research questions. First of all, what is the nature of the relationship between population size and contact rates in checkerspot butterfly larvae? And second, do observed patterns of habitat use lead to contact rates that differ from the null expectation of random habitat use? While population density is a better predictor of contact than population size, the contact process hinges on host movement. Checkerspot larvae form gregarious webs or nests on their natal host plants. And this is because the females are laying large batches of several hundred eggs at a time. As the larvae grow, the later instars disperse and forage independently. It is during this foraging dispersal that the majority of JCDNV transmission is likely to occur. So getting into the methods of this study, first of all, we have nest location, so where all those webs were located. And for this, we have larval nest occurrence data from 6 sites and 16 site years, so multiple years for all of the sites. Sites were systematically surveyed by past Crone lab members between 2013 and 2019. And here's just to demonstrate the type of data that we have for this. The number of nests varies, and some of these sites are in Maryland and some are in Massachusetts. And in Maryland, all of these were plantain feeding populations, while in Massachusetts, it was more, actually I said that backwards. In Maryland, it was mostly turtlehead feeding populations. In Massachusetts, it was a mix of plantain and turtlehead feeding populations. And I'm going to point out one site in particular, because later in this talk, I'm going to drill into this in a little bit more detail. So this is the Upton site, and this is just a demonstration of the pattern of spatial use at low and high populations levels. And then the other type of data that's incorporated into this are larval movement tracking. So during the dispersal phase of the larval growth, I went out and I tracked individual movement paths, so 98 different paths, and each path consisted of following a larva for 20 minutes apiece. And then from these paths, I discretized the larval movement, and in the discretized paths, I estimated mean step lengths, step times, and turning angles for each dispersing larval instar. Then following Kareva and Shigesada from 1983 and Turch in 1998, I calculated a mean diffusion coefficient for each instar. And then for this analysis, I'm just drawing on the 5th instar, partially because I think it's epidemiological most relevant for this system, and also it's kind of just a nice intermediate stage to look at these patterns. So with these diffusion coefficients, combined with field observations of how much time larvae are actually spending moving, I was able to come up with dispersal probability distribution functions. And this is essentially just a 3-dimensional bell curve. And here you can see 2 of these that represent 2 different nests. And the overlap where they're intersecting is showing how much expected contact we would have between 2 nests at a given distance apart. And then using the nest locations from each site in a given year, I generated pairwise distance matrices for that site and year, and then used the Shaner index to calculate overlap in the probability distribution functions between each pair of nests at a given site. And this gave the total expected contact for each nest in a site year. So just to review the research questions, what is the nature of the relationship between population size and contact rates in checkerspot butterfly larvae? And so here, you can see that if you plot the number of nests, which is a proxy for population size on the x-axis, and then the mean per nest contact, we end up with an asymptotic relationship between contact rates and population size. So many of the smaller populations actually had an estimated high level of contact, and that increases with population size to a certain extent, but not in a linear fashion. I also want to just point out, so this really points to site-specific habitat characteristics playing a large role in determining how population size and contact interact. In general, sites with lower population sizes had greater contact than would be predicted by a non-spatial disease model. Then for the second research question, do observed patterns of habitat use lead to contact rates that differ from the null exploitation of random habitat use? I used this Upton site Partially because there's a picture of it on the left. It's just a rectangle, so it's pretty easy to generate random nests at kind of randomly spaced nests, hypothetical nest locations here. And I should point out that Plantago, the primary host plant at the site, occurs throughout that whole field. So on the top, we see the observed distribution of nests. And then the bottom, and then n equals 44 was the side when there were 44 nests detected. And then n equals 309 was another year where there were 309 nests detected. So low and high population sizes there. And then on the bottom, I just randomly generated nests across the site. And then I estimated what the contact rates would be at these, and observed nests have a substantially higher contact rate than randomly placed nests in the same habitat at both population sizes. This is important because it will lower the population size required to maintain pathogen persistence, or R0. It also may lead to stronger density-dependent disease feedbacks than would be expected based on non-spatial disease models. However, I should note that in this case of a single site over a range of population sizes, there does appear to be a linear trend between population size and the estimated contact rates. Together with the results of this across-site analysis, this suggests that knowing only population size isn't sufficient for predicting contact. So conclusions, in the all-site analysis, there's an asymptotic relationship between population size and the estimated host-host contact, with higher than expected contact in small populations. In the random versus observed contact analysis using the Upton site alone, at every population size, the observed nest occurrence had a much higher contact rate than randomly placed nests. And overall, shared host habitat preference leads to substantially higher rates of contact than would be predicted by non-spatial disease models. This in turn will lead to greater than expected transmission rates, and thus a lower population size required to sustain a pathogen. Then finally, in plantain using populations, spatial patterns of host plant quality seem to play a role in larval distribution and the resulting contact rates. This is inferred from the presence of unused patches of plantain across the site. However, at this point, we don't have data that informs what is driving female oviposition preferences. Here's some acknowledgements, especially to the Smilanich lab out in Reno, and Lee Brown, June Oriens, James Michelini for collecting the nest location data used in this analysis. All right, thank you very much.